Welcome, everyone. Today, we will study mass spectrometry, an important analytical technique used to determine the molecular weight, structure, and composition of compounds. It converts molecules into ions and measures their mass-to-charge ratio, also called m/z. The first step in mass spectrometry is ionization. During this process, sample molecules are converted into charged ions. This is important because only charged particles can be manipulated by electric and magnetic fields within the instrument. The second step is acceleration. The generated ions are accelerated using electric fields, giving them approximately the same kinetic energy before they enter the mass analyzer. Next comes separation. In this stage, ions are separated according to their mass-to-charge ratio. Ions with smaller m/z values are deflected more than heavier ions, allowing different ions to be distinguished from one another. The final step is detection. The separated ions strike a detector, which converts their impacts into electrical signals. These signals are processed to produce a mass spectrum. It is important to distinguish between mass spectrometry and spectroscopy. Mass spectrometry measures the masses of ions and produces a mass spectrum based on m/z values. In contrast, spectroscopy studies the interaction of matter with light and produces absorption or emission spectra based on wavelengths or frequencies. Mass spectra are extremely valuable because each compound generates a unique fragmentation pattern that acts as a molecular fingerprint. This allows scientists to identify unknown substances accurately. For example, benzene shows a molecular ion peak at m/z78, while ethanol exhibits a molecular ion peak at z46, along with characteristic fragment peaks. To understand the importance of mass spectrometry, it is useful to look at its historical development. The journey began in 1918, when A.J. Dempster built one of the first mass spectrometers. This instrument separated ions according to their mass-to-charge ratio using magnetic fields. His work laid the foundation for isotope identification, atomic mass measurements, and molecular analysis. During the 1940s, mass spectrometry entered the stage of commercial development. Commercial instruments became available and were primarily used in the petroleum industry for hydrocarbon identification and industrial chemical analysis. These instruments helped scientists determine the composition of complex hydrocarbon mixtures more efficiently. In the 1960s, mass spectrometry expanded significantly into organic chemistry. Major technological improvements included the development of electron impact ionization, better vacuum systems, advanced magnetic analyzers, and more sensitive detectors. These advances enabled accurate determination of molecular structures and greatly increased the technique's analytical capabilities. In the modern era, mass spectrometry has become an essential tool in many scientific fields. It is widely used in pharmaceuticals, proteomics, environmental analysis, forensic science, food and agriculture, and materials science. Modern mass spectrometers can measure masses with extremely high accuracy, often within a few parts per million, making them among the most powerful analytical instruments available today. Mass spectrometry has many important applications in organic chemistry. First, it helps determine the molecular weight of a compound through the molecular ion peak. For example, methanol gives a peak at m/z32, while benzene gives a peak at m/z78. Second, it helps determine the molecular formula. High-resolution mass spectrometry provides exact mass measurements and isotopic patterns that reveal elemental composition. Third, fragmentation analysis provides structural information. Molecules break into smaller fragments, and these fragment ions help identify functional groups and bonding patterns. Mass spectrometry is also used for structural elucidation. When combined with IR, NMR, and UV visible spectroscopy, it helps determine the complete molecular structure. Finally, it is useful for quantitative analysis, as ion intensity is proportional to concentration, allowing accurate measurement of compounds. Because of these advantages, mass spectrometry is widely used in drug analysis, environmental monitoring, clinical diagnostics, and food quality testing. So, mass spectrometry is not just a machine for measuring mass. It is a powerful tool that helps scientists identify unknown compounds and understand their molecular structure clearly.